Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I have with me Phillip L'Huillier, CEO of Scancell, for a discussion on the broader oncology market and outlook. Welcome, Phillip.
Hello, Nicole. Thank you for the opportunity to join you on this podcast.
You're very welcome, Phillip. So, Scancell develops novel immunotherapy products for the treatment of multiple cancers, and it has recently announced a partnership with the NHS Cancer Vaccine Launchpad, otherwise known as CVLP, to fast-track access for NHS patients into the fourth cohort of the company's phase 2 clinical scope study in melanoma, to which we'll return in a second with updated information. As most listeners will realise, melanoma is the fifth most common cancer in the UK, accounting for around 4% of all new cancer cases. While immunotherapy is a standard treatment for advanced melanoma patients, only about half of patients respond well, leaving the rest at risk of disease progression and metastases. And last week, Scancell reported data from the phase 2 scope trial of SCIB1. But before we properly begin discussing all of that, Phillip, perhaps you could share a little bit more about yourself with listeners and describe the passions and the journey that took you to doing what you do today at Scancell.
Well, yes, thank you, Nicole. Let me tell you a little about myself and then also a little bit about Scancell before we jump into a more detailed conversation. I'm Phil L'Huillier, CEO of Scancell, and I guess I would say first and foremost, I'm a biologist with a strong interest in biology, in the beauty of nature, and that's what took me into science and then took me into research. And I'm now, I would say, a successful builder and leader of biotech companies, both here in the UK, but also in Europe. I started RTS Pharma with Kate Bingham from SV a number of years ago, Cioxis also, and a number of other companies. In more recent times, I joined US Merck & Co. in 2017, and I specifically joined Merck to gain late-stage development experience, clinical development experience in the late stage and in the commercialization stages, which is now very useful for me where I am at Scancell, and we'll come back to that. I was also part of the BD organization there, and that was an interesting perspective. I had spent years, as I say, selling into pharma cancer opportunities, and the Merck experience gave me the opportunity to be a buyer, and I led on a number of acquisitions. From Merck in 2021, so after 4 years there, I actually took over leadership of a Munich-based immuno-oncology company, Catalent. Catalent has assets in the IO space, in a number of cancers, and we, as a team, developed the lead asset from phase 1 through to randomized studies in phase 2B over a 3-year period, and raised $200 million to develop those assets. I joined Scancell a little over 6 months ago now, so a relatively newcomer to the company, and I saw a very strong pipeline of assets, really good deep immunology and translational science, as well as clinical capability, and a strong team. And that's what attracted me to join Scancell. Just a little on Scancell, perhaps. It's a UK public company. It's focused on the development of immunotherapies. We describe our mission as immunotherapies for a cancer-free future by enhancing long-term tumor immunity. We're clinical stage with, as I mentioned, deep roots in cancer immunology and also translational research. And Scancell develops off-the-shelf targeted immunotherapies with highly favorable safety profiles and durable long-term immune responses. The lead program, as you touched on, Nicole, is from our ImmunoBody platform. iSCIB1 is the first generation product, and iSCIB1 Plus is the second generation product. And we shared, as you said, a few results from the studies with these two products last week. They're showing really strong promise on top of checkpoint therapies, which are the standard of care in the UK and elsewhere in the late stage, in the metastatic melanoma. Scancell also has a second product. It's called Modi-1, coming from the Moditope platform. So the company has two platforms that has created these lead products, but also other products in the pipeline. And Modi-1 is being investigated in head and neck cancer and renal cancer at the present time. It has a broader application across multiple solid tumor types, whereas the iSCIB1 Plus product is a very specific melanoma product. We also have a set of antibodies targeting glycans, the sugars on tumor cells, as preclinical assets. And we've recently taken these preclinical assets and the capability and the platform into a new company, a subsidiary company called Glymab Therapeutics Limited. Why we've done that is to bring in investment specific to the stage of development, but also to those antibodies to move them forward more quickly into the clinic, and also to bring the right focus to the clinical stage part of the company, but also the preclinical stage part of the company. So that's a little about myself and Scancell.
Thank you for sharing all that, Philip. So I love this notion of your sort of entry into science coming from the biologist's angle and the deep appreciation of the beauty of nature. And then if we sort of segue to where you've been with Scancell just over 6 months now, and its goal being this cancer-free future, let's shift to that, if you will, with the partnership with the NHS. So if we return to the CVLP, the launchpad mentioned before, the NHS Cancer Vaccine Launchpad, if you will, launched in May last year as a world-leading NHS trial matchmaking service, and I'll put quotes around the word matchmaking, to help find new life-saving treatments. And the partnership is being coordinated and run by the Southampton Clinical Trials Unit, and Scancell has been the first British company to join the CVLP. So I was wondering if you could tell us more about how that partnership came about.
Yes, happy to, Nicole. And you stole my thunder. I wanted to tell you that we were the first British company to be selected to work with the Cancer Vaccine Launchpad. And I actually see that as quite a significant milestone for Scancell, of course, but also for UK innovation and UK melanoma patients. It's a real endorsement of what we're doing with our experimental medicine to be able to partner with the Cancer Vaccine Launchpad. As you said, it's a highly innovative model, and I agree, a collaboration between the NHS England, Genomics England, NIHR, and being led out of Southampton. That's quite a unique model that brings together the hospital structure. It brings together genomics capability to characterize patients at a genomic level. And it's a great combination for a study like ours, where we do need to type our patients and then bring patients in that are the right type to the study. And after conversations with the Cancer Vaccine Launchpad, they felt that our study fitted the types of studies that they want to focus on. And for us, it's important because it gives us broader access to patients, a broader access to the hospital structure, so to speak, across the UK. Our SCOPE clinical trial that is partnered with the NHS Cancer Vaccine Launchpad is being run. We run it out of 15 centres in the UK, and the Cancer Vaccine Launchpad enables us to access potentially up to 50 hospitals. So we're bringing more melanoma patients forward, able to screen those patients and then bring them into our clinical centres if they meet the criteria to enter the study.
Thank you. That's quite a considerable sort of pool of possibility with the hospitals there. 50 for your 15 centres for the SCOPE trial that you were mentioning there. So if we turn to the clinical SCOPE study now, and the phase 2 cohort evaluated intradermal administration of Scancell's iSCIB1 Plus, potent targeted off-the-shelf immunobody second-generation DNA cancer vaccine in patients, as we've been discussing, with advanced unresectable melanoma receiving standard of care immunotherapy treatments. So I was wondering, Philip, if you could tell us more about the sort of foundations of that study, the development of this DNA cancer vaccine, and also, of course, the new data from last week from the SCOPE study, in terms of how this is potentially going to transform the immunotherapy landscape?
Let me first of all describe some of the key attributes of the therapy, and I'm going to focus on iSCIB1 Plus because that was part of the decisions that we made last week. But iSCIB1 Plus, first and foremost, is an off-the-shelf therapy, meaning it's readily made, doesn't require the cost and time, a process of customization for each patient, and it targets its shared antigens that are common to melanoma patients. And in fact, the two shared antigens in the therapy target TRP2 and GP100, which are both proteins involved in melanin production. So this is very much a melanoma-specific vaccine. Because we've got these shared antigens there, then we don't need also the tumor sequencing process, neoantigen prediction, individualized therapy productions. So those are time and cost intensive steps to make personalized therapies. We are non-personalized, off the shelf. So that's the first thing to say about the therapy. The study, the SCOPE study, we describe it as a translational study. We're asking questions around which product of our two products will be the right product to move into later development, which patients will be the right patients to include in future studies. And then, as you touched on, Nicole, we're evaluating intradermal delivery versus intramuscular delivery, and also evaluating different immunization regimens. So there's quite a number of questions we ask across several cohorts in this study. And then additionally, we are also comparing checkpoints, the Nivo and Ipi combination versus the pembrolizumab as a standard of care therapy. So there are arms of the study asking different questions. But importantly, the purpose of the study is to answer these questions about which product, which patient group, and how do we deliver our therapy. We're running this across the centres in the UK. We've fully recruited the first 3 cohorts in the study, and the fourth, that's the one that's in partnership with the Cancer Vaccine Launchpad, is currently recruiting, and it's recruiting nicely. We announced an initial set of data end of last year for cohort 1, and then last week, as you said, a further set of data from cohorts 1, 2, and 3. And I expect we'll have data from this 4th cohort towards the end of the year. What I'm really, really pleased about from our data is we said to the market that we would come back at this time of the year and make a decision on which product we want to move forward with for registrational development. And we've been able to do that. The 2nd generation product is showing really strong efficacy in the target population, and responses look very durable. It's early days on the durability. Patients are still on therapy. We're still monitoring the durability, but we're seeing efficacy, durability, and not seeing any safety issues being added on top of the checkpoint therapy. So our therapy is safe and can be added on to other therapies, which is a nice feature for our therapy. We saw activity with both the pembrolizumab arm, but also, of course, with the Ipi-Nivo arm. And one of the nice things about the data that we've just announced is the 2nd generation product was designed to work in patients of particular HLA haplotypes. It was a set of class 1 HLA haplotypes, and we're in fact seeing in those patients, we're seeing out really strong efficacy. So I'm very pleased about that. The molecule was designed to work in a particular way. It showed up preclinically to do that, but now it's starting to emerge clinically that it's working in the way that it was designed. A lot more data to come. We haven't got all of the data for our patients yet. We've just shared a snapshot of the data at this stage. There's about, I think, 7 patients that haven't reached their first scan at this stage, and we've got more patients to come, but it's looking very, very positive.
Very positive indeed. So let's look to the future finally. And with this new data, this positive outlook, let's go even more generally. all things considered and with the possibilities of this, where is immuno-oncology heading, and what would be your hopes for the field in the next decade, Philip?
That's an interesting question. Perhaps, firstly, I would say we have developed a much deeper understanding of tumor immunology, including how tumors evade immune detection and how the immune system can be reprimed to recognize cancer antigens. So it's a pretty pivotal time from the early learning and from the breakthroughs. Of course, immunotherapy and checkpoint therapies have made a massive difference across many tumor types, not all, but many tumor types. And an example, which we were really just touching on, melanoma, survival in melanoma, 5-year survival in melanoma before immunotherapies was really very, very low. And the checkpoint therapies have made a difference, but there's a lot of work to do. I think the exciting work that's going on now that we'll see come forward is the breadth of modalities. We're now adding conjugate molecules, ADCs, radioligand therapies on top of the checkpoint therapies. We're adding our cancer vaccine therapy on top of checkpoints. So there's a lot of work going on different combinations, and that's the way the field is going to go. I'm pretty sure that we'll see many more combination therapies. Combination therapies where the combination is given at the same time to the patient, but also combination therapies that are sequenced. You have one therapy and then you move on to another therapy. And on top of ADCs and cancer vaccines, we're also starting to see T-cell engagers starting to make a big difference. There's already a number of indications where T-cell engagers have reached the clinic and are being evaluated in the clinic, bispecifics as well as other forms of molecules, again bringing benefit as single agents or as combinations. Perhaps the last piece that I think needs to be said is we still need to do a lot of work on identifying the patients that respond the best. We're still using an approach where we treat many patients, but only a portion of those patients see any substantial benefit. Billions of dollars has been poured into research to try to identify biomarkers. We're not there yet. Some progress being made. But that's another important area, really being able to hone in on the patients that respond to the therapy, and then we treat those patients with the therapies. And then also, of course, from that understanding, we'll be able to work out better for the patients that are not responding, what do they need to be able to respond? So exciting, really exciting times, a lot of progress, but a lot of questions and further work to do.
Absolutely. Thank you very much for your time today, Philip. It's been a pleasure.
Thank you, Nicole.
And that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on Apple Podcasts, Spotify, Overcast, Pocket Casts, Podbean, and pretty much wherever else you download your other podcasts from. Just search for pharmaphorum and follow and hit subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us on Blue Sky and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.